All right, thanks very much. It's my pleasure to introduce Mike Gregory, our next speaker today, and give an introduction. And what Mike's going to tell about is an application of this Nanodisc platform that we have that we've invented in our lab over the last many years. Have been trying to apply it for a variety of different systems. And the beauty of the Nanodisc system, as you can see there, is it is true nanoscale. It's basically a phospholipid bilayer that self-assembles in a magic process, whereby it is rendered soluble in aqueous solution because there's a circular purple belt that is amphipathic peptide that we designed that's nonpolar on the inside that interfaces with the alkyl chains of the phospholipid bilayer and is polar on the outside that renders it soluble in aqueous solutions. And the magic, of course, is that you can now put any kind of membrane protein that you want into this Nanodisc system and now have an ability to study membrane proteins in a wide variety of contexts, including, for example, interfacing them to a variety of different nanoparticle-based sensor modalities. So as an example of some of the success stories in terms of cancer targets and other potential applications for therapeutic intervention, we can put G-protein-coupled receptors into nanodiscs. We can put complex signaling molecules like a trimer of dimers involved in chemotactic reception signaling. We can put complex machines like the SEC translocase into nanodiscs, cholera toxins, various P450 systems that Mike will talk about, which are important drug targets for prostate and breast cancer. We can look at blood coagulation effects or any kinds of systems which are actually catalyzed by interacting with the membrane surface, and a variety of other different kinds of systems. You can actually put also machines into these by making larger nanodisc structures. We've put things like the full cytochrome oxidase complex shown there on the left. We've put things like the photosynthetic reaction center into these nanodisc systems and interface those with things like carbon nanotubes. You can put little motors in like the F0, F1 ATPase, affix that to a surface, use the nanodisc system to immobilize the membrane protein component, and then watch motion of the ATPase as it undergoes ATP-driven mechanical motion. SEC translocase I mentioned. This is an example in the middle there, I guess, is the ribosome bound to the SEC translocase inside a nanodisc. That's a real live three-dimensional cryo-EM reconstruction. You can actually even see the peptide that's being made by the ribosome come through the nanodisc system and protrude on the other side of the phospholipid bilayer. And the lower picture is a cholera toxin that's actually bound to a nanodisc structure without the nanodisc being terribly well defined in that particular cryo-EM. You can use this to do structures, which gives you a basis for rational-based drug design, which Mike will tell you a little bit about. If you're going to do structures, then you need to have a wide variety of tools that you can bring to bear on this, and there's been a large number of publications recently using solution NMR, solid-state NMR, calculations based on theory, atomic force microscopy, even crystallography, using the Nanodisc platform as a way of immobilizing and studying membrane protein structures. Sensing modalities has been a big interest in our group, and the nice thing about the Nanodisc system is that you can use it as a cassette to interface, again, any membrane protein target you might want with a variety of different sensor modalities, including some in the upper left here. Some localized surface plasmon resonance measurements where you look at electronic coupling between the inserted chromophore in the nanodisc and a localized surface plasmon that's in a gold nanoparticle. You can do surface-enhanced Raman spectroscopy, do normal SPR-based systems. Cantilever detection also works using the nanodisc, again, to interface with a mechanical sensor modality with a particular membrane protein. Mass spectrometry, sorting with a variety of different fluidic or gas phase sorting also seems to work. In terms of delivery, I put this in for Steve to know that in fact you can actually do delivery with this. Here's a tail vein injection of a nanodisc that's loaded, in this case with 10 molecules of cyptate as a far infrared imaging dye, and you can look at the localized and watch it come back and excrete out of the liver in the standard sort of clearance modality. You can measure plasma lifetimes. One of the interesting things, because these are so small, they do get into the microvasculature, and they actually seem to also cross the blood-brain barrier, which is another interesting thing that we have not had a chance to explore to this date. So we think this is a nice system for sort of looking at answering a variety of different questions that are related to cancer, and Mike will tell you a little bit about one application in prostate cancer.
Thank you, Steve. Afternoon. So today I'm going to tell you about some of my work with CYP17, which is a steroidogenic P450 that's been kind of interest in this enzyme has been growing lately as a result of prostate cancer and development of certain inhibitors of this drug. So I'd like to frame this talk first with a brief discussion of cancer. So the two most common types of cancer in the U.S. are prostate and breast cancers, and these have come to be known as sex hormone-dependent carcinomas, and that's because 80 to 90% of prostate cancers proliferate in response to androgen receptor activation, and in the case of breast cancers, roughly 70% respond to ER activation for their proliferation. And what this gives you, this dependence on sex hormones, is an inroad into treating these diseases through control of this steroid hormone biosynthetic pathway. So shown right here is the first committed step of androgen biosynthesis. So you have DHEA, androstenedione, and then a second aromatization reaction by CYP19 forms these estrogens. So what I'll talk about today is CYP17, which lies right here. Speaking generally, I could spend a half hour going through this pathway. But all these hormones come from the same precursor, which is cholesterol. This side chain is clipped off by CYP11A1 side chain cleavage, which gives you pregnenolone. It's also easily converted to progesterone. And then CYP17 comes in here, and it performs a 17-alpha hydroxylation on this pregnenolone and progesterone. And a second reaction in the same enzyme, CYP17, in the presence of its allosteric effector, cytochrome B5, results in 1720 carbon-carbon bond scission, which is the first committed step of androgen biosynthesis. And then later, of course, with CYP19, you have these aromatization reactions, which give rise to your estrogens. These two pathways have been studied for quite a while, and lately, in recent years, I'd say, the pharmaceutical industry has really capitalized on their role in the proliferation of these malignancies. Anastrol, letrozole, exemestane, these are all aromatase inhibitors. They came out beginning in the mid-90s, and more recently, abiraterone acetate, an inhibitor of CYP17, has just been released last year. So while these are great first steps, I think that they're no magic bullets. In the case of abiraterone, it's not a cure, I think it's worth saying. Typically, patients with advanced prostate cancer treated with this drug live an additional 6 to 9 months after beginning treatment relative to placebo. It also has some pretty severe side effects. You knock out this hydroxylation activity, and then you get an excess of aldosterone. It throws off your whole homeostasis, and you have to watch for hypertension, fluid retention. And it's kind of a nasty thing. So I think the defining question within probably the medical community and the scientific community is, can we do better? But in order to do that, I think you have to have a fundamental understanding of this steroidogenic pathway. And in order to gain that understanding, you have to understand the cytochromes P450. You can see each dot here represents the activity of a cytochrome P450. So what are they? The cytochromes are a large superfamily of enzymes that are generally regarded as the hydroxylators of unactivated alkane chains. It's a huge superfamily, absolutely ubiquitous, present in all branches of the tree of life. In 2009, there were 11,000 sequences, and today I think it's over 17,000. They share a similar protein fold, and the key to their interesting catalysis is this heme located in the active site with cysteine proximal ligand. We humans have 57 of them. They do lots of things, but most important to the medical community here is a role in xenobiotic metabolism. About 70% of drugs are worked on by a P450, and as you saw before, steroid hormone synthesis. Moving back to this talk of inhibitors, it's not that hard to develop something that will, or to find something that will inhibit a P450. All you have to do is coordinate something to this heme iron, and that'll ablate activity. The question is specificity. If you knock out all 57 P450s in a human, cancer's only one of your now problems, because you're in trouble. So you really have to target your chemistry very closely. And the way this has been done in the past is through substrate recognition events. All these inhibitors that I showed you before are recognized by the active site, and only the certain molecules will fit in. And it's pretty good. In the case of abiraterone, it's very specific to CYP17, but it also inhibits CYP2D6, so you have to watch what other drugs you take. But I think the question again is, can we do better? Is there another way that we can target these? Maybe a mechanism-based inhibitor. But the problem with P450s is that, in general, the answer to that question is no. You have this dogmatic catalytic cycle that's generally true throughout this whole superfamily. And just briefly, I'll walk through this. You begin with this ferric resting enzyme. You bind substrate, and then your iron can be reduced to the ferrous form. Then you combine molecular oxygen here and form the oxy complex. This is the last stable intermediate. or quasi-stable intermediate. Second reduction forms the peroxoanion, and you protonate this to form the hydroperoxo. A second protonation of events results in heterolytic scission of this dioxygen bond and formation of the archetypal reactive intermediate in P450's compound 1. And what this does is abstract a hydrogen off of an alkane, or off of a carbon, which is followed by oxygen rebound, giving you your hydroxylated substrate, which floats off, and you're back to your resting enzyme. So this is true of, in general, I think, of the overwhelming majority of P450s. But in the case of CYP17, it's not clear that that's what happens. So you can draw several reactions that progress through this hydrogen abstraction. Maybe you rip something off this hydroxyl here on C17. And this is the lyase chemistry of hydroxypregnenolone, the first committed step of endogen biosynthesis. And maybe you can rip something off of here and go through radical chemistry. But about 20 years ago, some people realized that, in fact, maybe this electrophilic moiety here, this carbonyl on C20, maybe this can direct this peroxoanion into a nucleophilic attack. And you can fragment this in a couple different ways based on whether you believe it goes through heterolytic or homolytic scission of this bond. But the bottom line is that this breaks down to form your androgen, just as something else would. And this debate has gone on now for 2 decades, but there's no clear answer to this question yet. There's some circumstantial evidence, I think, but there's no answer yet. So I think the question that we ask is, how do we get to this? But there's a problem. One is that CYP17 and all human P450s are membrane proteins, which means they're insoluble. Sure, you can add in detergent and they'll float around, but the problem with detergent solubilization is that oftentimes you get these aggregated non-native conformations. And some of the physical properties that they'll display are not necessarily physiologically relevant. You can't control the aggregation state of these, so it makes it very difficult to do reconstitution assays with controlled stoichiometry. And also very important is that this membrane bilayer, it's often a very important structural element in maintaining the native fold of membrane proteins. So without it, they'll fall apart. Like I said, you can't control stoichiometry. And another thing, oftentimes in detergents, the substrate molecules will partition preferentially into the detergent micelles. So your KDs and KMs that you pull out aren't representative, perhaps, of what you'd see in the body. So the solution, as Steve told you, is nanotechnology. Steve already covered this, so I won't dwell too long on this. But like he said, you have this soluble lipid bilayer that has your target protein embedded. And like he said, you can get a monomeric protein. You can put in other proteins with it in controlled stoichiometric ratios. And they're robust, monomeric. The way that we make these, you begin with your detergent-solubilized target protein. We obtain this from heterologous expression. You take your membrane scaffold protein, which forms this belt here, and detergent-solubilized lipids, and you combine these all in precise ratios, and then you pull out this detergent with hydrophobic beads, and these whole things self-assemble. And what you'll get if you control these stoichiometries properly are 2 populations, one containing disks with target and one full of empty disks. And then you can isolate this fraction through use of an affinity tag on your target, and then you go through a second step of size exclusion chromatography, pull out the appropriately sized fractions, and now you have your disks. And also, since Steve mentioned this, I'll just breeze through this slide, but this enables a whole lot of stuff. You can do lots of solution-based assays, spectroscopies, reconstitution assays, product formation, the laydown flat on mica, you can do AFM. Here's a biotinylated MSP on a photonic crystal biosensor, and we're sticking annexin to anionic lipids. So there's a solution to the first problem. The second is, what experiment do you do to find out what reactive intermediate is giving rise to this lyase chemistry? And a defining feature of this later phase of the catalytic cycle is that if you begin here with this ferric peroxoanion and you go straight to product, you don't need to invoke the use of any protons. But if you move along to compound 1, the archetypal reactive intermediate, you have to have at least 2 protons in flight. So what we can do is look at kinetic solvent isotope effects, beginning with this ferric peroxoanion. The way that this works is that you cryotrap this species at 77 Kelvin. You put this into an optical cryostat that we have here, and then you linearly ramp the temperature, and eventually you put in enough energy into the system to get over this barrier, and then the reaction progresses as follows. This is, say, percent or fraction peroxoanion. If you exchange buffer of your proteated solvent for deuterated solvent, and you're going through compound 1, since you have these protons, you'll see a lower 0-point energy, higher energy barrier. You'll see a right shift of this reaction profile. In the case of going through the ferric peroxoanion, there are no protons involved, so you'll see two traces like this in your H2O and D2O. And the way that all this works is that you begin, this is the experiment that we do here, you start with your ferric enzyme, add substrate, chemically reduces dithionite, then you bubble in oxygen at low temperature, say minus 30 Celsius, so that this is stable enough. Then you freeze to at 77 Kelvin, irradiate with gamma radiation, which provides, among other things, solvated electrons, which reduce this oxycomplex to the peroxoanion. And then you put it in the cryostat that I showed you, and you can watch the characteristic optical spectra of this peroxoanion decay as a function of temperature. And when you do this, you get these traces, like I showed you before. This is annealing the peroxoanion of pregnenolone. So this is a standard P450 mediated reaction going through compound 1. It's just a standard hydroxylation. And as you'd expect, in H2O you get this profile, and it's right shifted in the presence of D2O. So that provides evidence of protons involved in compound 1, as expected. In the case of annealing the 17-alpha hydroxypregnenolone peroxo intermediate, and this is the questioned and hotly debated mechanism, you in fact do not see a KSIE. So here is H2O and that's D2O. They overlap. So this is evidence of no protons involved and going through this ferric peroxoanion, which is really novel. Among the P450 superfamily, this is pretty unique, and it is definitely unique among the 57 human P450s. So this kind of gives you a unique way to approach designing these drugs with for CYP17 inhibition. Perhaps you can utilize this unique chemistry that this particular enzyme directs this iron species to. Oh, it's also worth mentioning here that you can't do any of these in steady-state turnover assay. This KSE work in steady-state turnover. You have to start with this particular intermediate, otherwise it's all masked. So, like I told you before, we're heading toward, we'd like to guide development of this inhibitor. So what we propose here is to begin with this detailed biophysical characterization, combine it with detailed biochemical characterization of the properties that give rise to this lyase chemistry. Here is a high-pressure bomb with a photonic crystal in it. We can blow apart complexes of cytochrome P450 CYP17 and B5 or CPR and learn things about the volume changes, things like this. And we hope to compile all this, the acumen of all that we've learned, and rationally design and guide future drug development. So that's where we hope to go with this. Just a brief conclusion. The absence of KSIEs for the carbon-carbon lyase chemistry of hydroxypregnenolone implicates a nucleophilic attack in contrast of the hydrogen abstraction commonly exhibited by P450s, and it's distinct. I'd like to thank everyone here for attention, and of course Steve Sligar, Brian Cunningham, everyone in the Sligar lab. Many thanks, and I'll take any questions if you'd like.
So how do you plan to use the photonic crystals, Brian, with this project?
Well, I'm interested in exploring the B5 CYP17 interaction. So like I showed you, and I kind of glossed over this in the first slide, but this lyase chemistry is entirely dependent on association with this B5, this cytochrome B5. And it's an allosteric effector, and we don't really understand fundamentally how this interaction works and why it guides this interaction. So we're thinking that if we can learn some things about it by blowing this apart in a high-pressure bomb, disrupting this interaction, we can learn about how it's, what important residues are making these contacts, and perhaps guide future development, with that regard as well.
So you're looking for technologies where you could study interaction.
Yes.
Where the photonic crystals come in?
Yes. Yeah. Yeah. The nice thing about the photonic crystal in a high-pressure cell is that you can't do this with SPR, so you have to prism couple this. And our pressure bomb is a stainless steel thing with a window here. And the path length here is like 3 centimeters to get from outside to in. And there are refractive index changes and things like this. So the photonic crystal really gives us a nice method of observing these changes in protein-protein interactions on a surface. And we've been able to record compressibility of buffers and things like that. So it works. It's pretty cool.
If you go on the previous slide, you can have more things. So the y-axis is change in fluorescence, so what are we looking at?
Oh, no, this is change in fraction of, this y-axis is a fraction of hydroperoxo remaining.
I see.
So this is derived from an optical spectrum, and you watch the change in this.
Okay. All right, no further questions. Thank you.